Welcome to the biology section of our practice MCAT questions. In this video, we're going to be going through questions 131 to 135. So first I'll show you guys the questions so that you can pause the video and attempt them on your own. Here's question 131, 132, 133, 134, and 135. Alright, now let's go through the questions together. In question 131, we are asked which of the following structures is most necessary for proper function of cardiac muscle. So we're talking about the functioning of cardiac muscle, which is most necessary for these, okay? So cardiac muscle does have, most of these, but which one would be the most necessary? So which one's the best answer? Proper functioning. Tight junctions, these hold cells together and they have a very tight hold. That's why they're called tight junctions. It's so tight and they're so close together that not even water molecules or other liquid molecules can go through. So they're kind of impervious to osmosis happening throughout the cells, so like between the gaps in between cells. Whereas desmosomes, these are things which hold cells together so that you have cells in a tissue held onto each other instead of them just floating away. But they have a little bit of a gap between them. So this is like a linkage between the cell membrane of two cells, but there's a gap between them. So water can go through in the gaps between desmosomes. However, of course, the things which hold the cells together is necessary, but we're talking about the functioning of cardiac muscle. Something very important would be gap junctions. Okay, so I would say C is the best answer. Option D is centrioles, which are things that are involved in mitosis and creating the spindle fibers that help pull chromosomes apart. All of these things are important at some point for the cell. So it could be during mitosis, it could be during just the cell being together in a tissue. They're important, but they're important for like all types of cells. We're talking about the proper functioning specifically of cardiac muscle, and what cardiac muscle has to do is contract properly to move blood throughout the heart and then push it out to the rest of the body as well. And so it's very important that this contraction is coordinated, and so the action potential has to happen very quickly and pass throughout the rest of the cells in the heart quickly at the same time so that they contract properly. So we don't have like what we have in neurons. We don't have a synaptic cleft. We just have gap junctions which propagate the ions in an action potential directly from one cell into another, and this is a much faster way of propagating action potential. So the most necessary for cardiac muscle function would be gap junctions. So C is the best answer here. In question 132, it says urine passes from the kidney to the bladder via which of the following structures? So we are talking about urine going from the kidney to the bladder. So which structure is responsible for that? These are all kind of related to the kidney and the bladder, these structures. So not other parts of the system, but you can kind of confuse the names and what they represent, especially the first two, urethra and the ureter. These are very easy to confuse. The urethra is the one that goes to the outside. Okay, it goes from the bladder and then it goes out to whichever reproductive organ we're talking about, male or female, and it goes out to the outside. So it takes it takes urine away from the bladder and to the outside when you pee. So that is what the urethra does. But we're talking about what brings urine from the kidney to the bladder to be stored in the first place. That is a ureter. That tube called the ureter, okay? Urethra is the one that goes to the outside. C, the renal tubule, and then D, the loop of Henle. These are both parts of the nephron inside the kidney. So they're responsible for adjusting the concentrations of things like ions, water, as well as other molecules that are in the urine. So these adjust the concentrations of those. And so they adjust the concentration and the components of the urine. But none of these are structures which are responsible for carrying the urine from the kidney to the bladder. The correct answer here is the ureter, option B. In question 133, it says in the blood, lipids are often transferred via lipoproteins, which are composed primarily of lipids bound to proteins. That's why they're called lipoproteins. So this helps to emulsify the lipids for transport. In the digestive system, fat emulsification is accomplished by which of the following? So the main question that we're asked here, in the digestive system, fat emulsification, how is it accomplished? Emulsification, what it means is breaking down like fat molecules into smaller parts and increasing the surface area available for digestion. So what happens is, due to this hydrophilic environment inside the digestive system and the fact that lipids are hydrophobic, because of the hydrophobic effect, they're going to clump together into big clumps, and therefore digestive enzymes can really only reach things on the outside, and it's hard for them to work their way to the inside, break down lipids properly, especially during the time in which the bolus is passing throughout the digestive system. You want to do it quickly, right? So what we want to do is break up this big fat globule into smaller globules, break those up as much as we can into smaller and smaller globules, so that we have a bunch of small globules, a lot more surface area available for the digestive enzymes, the lipases, to act upon. That's what fat emulsification is. And how do we do that? It's not the lipases which are responsible for it. These are enzymes which break down lipids. Sure, they're very important for the digestive process in regards to lipids, but they're not the part that's actually related to the emulsification. So B is the correct answer. Bile salts. These are things which have both a positive and a negative side to them. And what they do is they form structures called micelles. So those would look like this. For example, if we have something where the circle represents a hydrophilic part, a hydrophilic head, and then the lines are the hydrophobic tails, you see that the hydrophilic part, the outside, is interacting with the hydrophilic environment, whereas on the inside we have a hydrophobic environment. So bile salts can kind of create this type of micelle environment, where on the outside the bile salt is interacting with the environment that's hydrophilic, and on the inside you have the hydrophobic parts of the lipids that are trapped there. If we have a bunch of these lipases, if we have a bunch of these micelles formed, and these are the small chunks that we're talking about instead of the big chunk of the fat globule, then these are going to prevent the parts of the lipid that are inside the micelle from reaggregating with the rest. And so what this does is it breaks down the fat globule, and it also prevents it from coming back together again. And then now the digestive enzymes have access to this, and they can start to break down the lipids. It's not stomach acid either. So digestion of lipids, it mainly happens in the small intestine, and it doesn't happen in the stomach. So it's not stomach acids which do this, and they're not able to do emulsification. You need a type of salt for this, not an acid. And then phospholipid, that's just a type of lipid with a phosphate group. So no, that's not something which helps for fat emulsification either. B is the correct answer here. In question 134, it says Ferrobacillus ferrooxidans is an organism that oxidizes this iron 2+ in this iron sulfide to produce sulfuric acid. What is the role of oxygen in its metabolism? So this bacteria is essentially, the main thing is it's oxidizing iron. It ends up producing sulfuric acid, but it oxidizes iron. And we're asked, what is the role of oxygen? If you're oxidizing something, you can use oxygen to do it. So that means that iron is being oxidized. You must have something that's doing that. So that thing that's oxidizing iron is an oxidizing agent. So oxygen, its role is that it's an oxidizing agent. Therefore, we can remove option D, which is saying reducing agent. It doesn't really make sense that oxygen would be a reducing agent, because we're just told that this bacteria, it oxidizes things. So it doesn't make sense to use a reducing agent. So oxygen is an oxidizing agent, and remember that if something is an oxidizing agent, that means that it oxidizes something else, and it itself in turn must be reduced. So it's not an electron source per se, because when something is reduced, that means that you have gained electrons. Option B is correct. It must have gained those electrons from the iron. That would be the electron source. It is the terminal electron acceptor. It has accepted electrons. Reduction is gain of electrons, and therefore oxygen got reduced, and it acted as an oxidizing agent. And C is incorrect. It's not a catalyst. Oxygen is not some catalyst, or it's not even Yeah, it's not some catalytic element in this reaction. What's being used for is part of a redox reaction, and it itself is something that gets reduced, whereas the iron, we're told, gets oxidized. In question 135, it says a sample of muscle tissue is noted to take significantly longer than regular muscle tissue to spread action potentials. After investigation, biologists deduced the problem is structural rather than chemical and is likely related to cellular membranes. Which of the following is the most likely reason for this problem? So think about how regular muscle tissue spreads action potentials, and now we have a small sample that takes significantly longer, okay? So think about how regular muscle action potential is propagated, and then now something in that process has gone wrong. We're told that this is structural rather than chemical, so there's some structure in the muscle tissue that is affected. It's not some chemical that's been affected. It's likely related to cellular membranes, so which one is the most likely reason? Option A is saying there's an irregularity with the size or existence of gap junctions. This one could make sense because that's how we have action potentials being propagated in muscle tissues. Remember, we're not talking about some neuromuscular junction. We're not talking about a neuron and it's propagating an action potential to a muscle tissue. We're talking about between muscle tissues, how they propagate action potentials. That would be using gap junctions, which are connections between different cells in muscle tissue that can directly transfer ions and propagate this action potential. So if there's a problem with them, it makes sense that now this subset of cells, the ones that we're looking at, the sample, it takes significantly longer to propagate the action potential. And it's a structural error, right? This isn't something chemical. Option B is saying there's a structural error in the desmosomes of the cell, causing excessive membrane elasticity. That doesn't really make sense. The things holding the cells together, there's a problem, so now they're elastic and not really held together as well. That doesn't make as much sense as gap junctions, which are actually directly involved in action potential propagation. This, if this happened, we'd see a lot of other problems rather than just the fact that action potentials take longer. It seems like everything else, the functioning of the cell seems to be fine. It's just the action potentials are taking a while to propagate. So it needs to be something which is more directly related to action potentials. Option C is saying there's an excess of tight junctions preventing the action potential from propagating. Similar to B, no. This would just hold the cells together more tightly, but we see nothing that kind of demonstrates that that is happening, and tight junctions are not directly related to action potentials. And finally, option D is saying there's a mutation in the gene for the membrane receptors which assist in propagating action potentials. And that's not correct because, like we discussed, we don't use membrane receptors in muscles to propagate action potentials. It is through the use of gap junctions. So A is the only one that makes sense. That's it for the questions in this video. If you enjoyed what you saw, make sure to check out our course. The link is right here on the page as well as in the description below. And in that course, we go through a lot more questions just like this and go through all the different answer options explaining why each one is correct or incorrect. Other than that, make sure to subscribe here to this channel to stay up to date with the videos that we post here. And that's it for this video. I'll see you guys in the next one.